Assalamualaikum everyone. I am Sehrish Gohar, PhD scholar from the Department of Biochemistry, ICAZ-2020 University, Islamabad. First of all, let me thank the organizing committee of ICAZ-2020 for providing me this opportunity of presenting my research work to the scientific community through their platform. I am going to present my research related to the contribution of mitochondrial calcium homeostasis to neutrophil extracellular trap formation. Neutrophils are the most abundant leukocytes in human blood that participate in various immunity alerts and inflammatory conditions through various mechanisms such as degranulation, phagocytosis, and release of their DNA in the form of extracellular traps known as nets. This nets formation is also known as netosis and is categorized into two main types based on the underlying mechanisms. First is the suicidal netosis that depends on the NADPH ROS to release the neutrophil extracellular traps after the lysis of neutrophils. This type of netosis leads to neutrophil death. The second type of netosis is vital netosis that is not dependent on NADPH oxidation and releases nets in the form of vesicles. This type of netosis does not lead to neutrophil death and hence the cell can continue its normal functions of phagocytosis and chemotaxis. Now, in the bacterial infections, the endotoxins and formylpeptides that are released by bacteria result in the oxidative burst, leading to formation of neutrophil extracellular traps. In this study, we have used a similar mimic of bacterial formylpeptides known as the formylmethylucyl phenylalanine that is known to bind the formylpeptide receptors on neutrophils and cause various neutrophil functions that are similar to bacterial formylpeptides. But the role of FMLP in the release of neutrophil extracellular traps has not been studied enough yet. It is controversial that whether this FMLP causes nets formation or not. Therefore, we designed this study to investigate the nets inducing potential of FMLP. Also, this FMLP is known to induce calcium alterations in the cytoplasm and mitochondria of neutrophils. There's a channel in mitochondria that is involved in the mitochondrial calcium homeostasis, particularly the uptake of mitochondrial calcium. This channel, mitochondrial calcium uniporter, is involved in neutrophil functions such as polarization and chemotaxis. However, there is no study about its involvement in the nets formation. In this study, we also focused on this channel to understand the contribution of mitochondrial calcium homeostasis to the FMLP-mediated nets formation. Usually, in the neutrophil function related studies, there is a density gradient based preparation which results in the isolation of neutrophils. However, after the density gradient based preparation, we found that isolated neutrophils were significantly activated without any treatment, and we did not find it reliable for our FMLP based stimulation of neutrophils. We used a close to physiological whole blood preparation containing white blood cells and red blood cells in a physiological buffer and found that the neutrophil morphology was close to normal in this preparation. So we used this preparation for our experiments and observed neutrophil morphology through light microscopy and quantified the release of cell-free DNA through these samples. After treatment with FMLP, we compared the percentages of normal and netotic neutrophils in the FMLP treated samples and found that the treatment of FMLP has resulted in more damaged neutrophils, releasing neutrophil extracellular traps. To verify our results of microscopy, we also performed quantification of cell-free DNA release and found that the FMLP treated samples were releasing more cell-free DNA. For the same samples, the pharmacological inhibition of mitochondrial calcium uptake resulted in the suppression of nets releasing neutrophils. You can see that the FMLP stimulated neutrophils have more netotic neutrophils, while the mitozentron, that is an inhibitor of mitochondrial calcium uptake, results in the decrease in percentage of nets releasing neutrophils. We also investigated the inhibition of complex 1 in mitochondria and found the same effect. To verify these results of microscopy, we performed the quantification of cell-free DNA and found that the pharmacological inhibition of mitochondrial calcium uptake and complex 1 resulted in a significant decrease in the release of cell-free DNA. The percentage of cell-free DNA as compared to control was lesser in both groups. To conclude, our results have emphasized the potential of FMLP to induce net formation at the very early stage of stimulation. Our data also supports the notion that mitochondrial calcium uptake contributes to FMLP mediated nets release. This study can help in understanding and hampering infection induced anomalous nets formation, which can instigate serious inflammatory diseases. Blocking mitochondrial calcium uptake can be a potential therapeutic target for the diseases caused by excessive nets released by neutrophils. In the end, I would like to acknowledge the continuous support and guidance of my supervisor, Dr. Mohammad Rizwan Alam, throughout the research. Finally, thank you for your patience and time